For this panel, we have, again, an eclectic group, but one that includes people who are thinking hard about how we can decide upon the rules and oversight mechanisms for the kinds of trials and information that will be needed to bring some of this information to the clinic. What are the hurdles and what are the needs? The moderator for this session is Michael Fischbach. Michael is an assistant professor in the Department of Bioengineering and Therapeutic Science at the University of California in San Francisco. Michael's work, in fact, I mentioned earlier this morning, involves a really interesting combination of chemistry and genomics. Michael is one of these people who is making fundamental discoveries into the kinds of small molecules that are made by the microbiota and that may have promise for possible future therapies. He's thought a lot about the translation part of this, and for that reason, he seems to be a perfect person to mediate this discussion. He, in turn, will introduce the others. Thank you, Michael.
Okay. Am I audible in the back? Good. Okay. You guys made it. This is the very last session. I've been told more than once that there's a cabinet secretary who's going to be speaking right after us. And I can't imagine why people might think that an academician like me might be undisciplined in timing. But this will keep us, in fact, quite disciplined. And so I'm going to keep my introductory remarks short. I have a couple of slides. We had agreed initially to not have any slides and to turn it over to questions as soon as possible. So I'm going to try to keep this quite compressed. It's a very exciting topic, and I think it's one about which there might be some lingering questions in the audience. Okay, first let me start by introducing the other people on the panel. To my immediate left is Billy Jo Hardy, who's a managing consultant in the Center for ESC Risk at the Li Ka-shing Knowledge Institute of St. Michael's Hospital, which is in Toronto. She leads a wide range of consultations. She works with almost everybody, including the Bill and Melinda Gates Foundation, and she received the Douglas Kinsella Doctoral Award for Research in Bioethics. So her research, rather than reading you the whole blurb, I'll just tell you that it involves a very interesting mixture of economics and ethics as it applies to genomics and other interesting emerging topics in biology. To her left is Scott Stibitz. I apologize if I got it right. Who is the chief of the Laboratory of Enteric and Sexually Transmitted Diseases, which is in the Division of Bacterial, Parasitic, and Allergenic Products in the Office of Vaccines Research and Review in the Center for Biologics Evaluation and Research at FDA. His research interests are in the general area of bacterial genetics and pathogenesis, with a special interest in some pathogens and some friendly organisms. And I think we're going to spend today talking, or this panel, talking mostly about the friendly organisms. So let me jump right in and just frame the discussion by saying that in my perhaps biased reading of where things are going, how we're going to turn the microbiome into something useful, there are three things people are thinking about. The first one of which I don't even have a slide on, but I'd welcome questions or comments from you guys or from the audience or from whomever about any of this. So I'll start off by saying the first one is diagnostics. Is it possible, for example, that we could diagnose Crohn's disease by enumerating stool, just from a stool sample that you would mail in from home, rather than having to go through a more onerous procedure and costly in the doctor's office? People are asking the same kind of question about, for example, colon cancer. So that's the first area in which people are thinking about translation. The second has to do with small molecule and biologic drugs that are produced by these bacteria. As many of you know, I'm putting the slide back up here that David showed you this morning, not to harp on it, but just as a cue for me to tell you that this is an interesting area. People are thinking about the fact that we've mined soil and marine bacteria for many, many of the drugs that are used in the clinic. About a third of all drugs used in the clinic come from microorganisms, and so people like me, it's self-serving for me to say it, but I'll say it anyway, have begun wondering whether there might be particularly interesting drugs that are naturally produced by the human microbiota, maybe by bugs that are present in some of us and not others, and that might be a neat place to look. That's all I'm going to say about that, because the third place people are looking, or the third kind of product that they're discussing developing, is in many ways the most interesting. So let me see if I can make it to that slide. If I can't, then I'm just going to have to use my words, which would be a disaster. Okay, here it is. So you've heard about this fecal transplantation study that garnered a lot of press, and for good reason. This was a study in which fecal transplantation was used to treat Clostridium difficile diarrhea. There had been many studies like it before, but this was the one in the New England Journal, and so it got a lot of people's attention. There were 3 points that I think this study made that were remarkable, and they go from remarkable to extraordinarily remarkable. So the first one that was just remarkable is the fact that it was so effective that they had to stop the trial early because it was so effective that it would have been unethical to keep people in the control arm. And that itself has been a sea change for treating Clostridium difficile diarrhea, and many hospitals, including the one at my research institution, have set up committees and new procedures for using feces as a treatment for Clostridium difficile diarrhea, something that we're going to talk about in a moment. The second and third things I would argue are even wilder and more impressive in terms of their implications for where this is going. The second one is that fecal transplantation, with a couple of minor exceptions, turns out to be remarkably safe. Much safer than you could imagine for any prospective small molecule or biologic drug. And that's something that I don't think could have been predicted ahead of time. Maybe some of you who know more about what had been happening ahead of this article would argue that it was known, but I would say that it's taken the world by surprise how such a profound, remarkable manipulation, replacing all of the gut bacteria with new ones, can be this safe. So that's huge. And the third thing, which is even huger, is the fact that there's no special gut community in you. That there doesn't need to be a relationship between the donor and the recipient, it would seem. So it's not yet clear whether that's a special case. Maybe patients with Clostridium difficile diarrhea are a particularly easy case, and it's particularly easy to get engraftment for the new community to come in and take hold. But if this turns out to be at all generalizable, then it's going to make developing therapeutics based on replacing all of the bacteria in somebody's gut quite an easy thing to do. And I would end just by saying, this is an article from a review that David and I wrote along with 2 co-authors, that if you think about the group of bacteria in somebody's gut as a network, the simplest thing you could do to the network is to very cleanly excise one problematic species, and that's something that there are vaccines that do that, and people, this morning we're talking about narrow-spectrum antibiotics to accomplish that. What is remarkable about fecal transplantation is how easy it seems that we can do the manipulation all the way at the other end of the spectrum, which is just to completely replace everybody that's there with a totally new community. So I would argue that that is unexpected, and that the fact that it seems so straightforward to do is something that could take the field in a completely different direction than people had anticipated even recently. So now I'm going to turn it over to Billy Joe for her introductory comments.
Thanks so much.